So I can go ahead. They have access?
Yes.
Okay. Welcome, good morning, actually good afternoon, everybody, and whichever time zone you're in around the world, zooming in here. It's a great pleasure to welcome Damian Grundle to today's special Global Futures Laboratory seminar. Damian will talk about the nitrogen cycle, actually nitrous oxide, part of the nitrogen cycle in the ocean, with the subtitle, "It's no laughing matter," with reference to laughing gas, of course. Just a little bit of background. Damian is an assistant scientist right now at the Bermuda Institute of Ocean Sciences. And his background is in biogeochemical oceanography. And his research right now is focused on nitrogen cycling with special emphasis on nitrous oxide, N2O. He's looking at the environmental impact on the production of nitrous oxide, what that means with respect to oxygen minimum zones, etc. And he will tell you much more about that. But also, and that was an interesting facet that I learned just recently, looking back at his work, how to measure how this gas, which is a greenhouse gas, many of you know that, is actually making its way from the ocean into the atmosphere. And these gas exchange measurements are pretty tricky to do. He's using the eddy covariance method. That reminded me of my work where we actually released two tracers and watched how they came back out into the atmosphere. Whichever way you do it, that's a tricky measurement. So I think he told me he's still making his way into that, but it's a very interesting facet of his work. Before joining BIOS, the Bermuda Institute of Ocean Sciences, Damian was a postdoctoral scholar and research scientist at GEOMAR, which is a research center for marine science in Kiel, Germany. And he got his education in his PhD in biogeochemical oceanography at the University of Victoria, Canada in 2012. So without further ado, I'd like to hand over to Damian.
Thanks, Peter. There we go. So yeah, thanks for everyone for coming along today, and thanks for inviting us out here this week. It's been an enjoyable morning and looking forward to more chats and tours around campus as the week goes on. So as Peter mentioned, I'm going to talk about nitrous oxide cycling in the ocean. First off, just as Peter mentioned, a little intro. I'm a biogeochemical oceanographer. This principally, from my side of things at least, includes sort of the study of the interaction between biology and chemistry in the ocean. One of the primary things I really focus on is the marine nitrogen cycle. And this is a whole suite of things. So it includes things like looking at phytoplankton nutrient utilization and how N cycling can be used to better understand carbon export via the biological pump. Right now, we're actually, I have an NSF funded project where we're using, with my co-PIs, Ruth Curry and Mike Lomas, we're using gliders to equip with a whole bunch of sensors, including nitrate sensors and microturbulent sensors to be able to sort of detect the upward movement of nitrate into the euphotic zone and better, better understand export production at much higher temporal resolutions than we could achieve by just simply going out once a month on a ship. I also look at oxidative versus reductive N cycling pathways. So this includes nitrification, which is the biological oxidation of ammonium to nitrate, and then denitrification, a reductive pathway, which looks at the reduction of nitrate to N2 gas and leads to a loss of bioavailable nitrogen from the ocean. But I mentioned one of the main things I look at is also nitrous oxide cycling in the context of nitrification and denitrification. And I'll come back to this in a second. Much more recently, I've also, with an amazing student that I had worked with me, Annalise Meyer, who's now in the MIT HUI PhD program, we were looking at rare earth elements in the ocean in relationship to methane cycling. And we recently just published what I thought was a pretty cool paper. I'm still learning a lot about this from Annalise, but recently just published this in Earth Planetary Science Letters. And we're able to look at rare earth element ratios to sort of understand what's been going on with methane in the ocean. But it also has some pretty cool sort of potential implications for things like astrobiology. Yeah, imagine if we can go off to a planet, let's say Mars, where we've detected methane. We can bring back soil samples and look at the rare earth element ratios, and we might be able to start saying something about whether this was due to biological activity or not. As I said, main focus of today is nitrous oxide seismic. So I'm going to start off with just touching on the importance of N2O, how N2O is produced, the implications of decreasing oxygen for N2O production in the ocean, give you some examples of past and more current sort of research activities, and then touch on some thoughts I have about future research. So first off, N2O is a climate relevant atmospheric trace gas, and the oceans are a significant source of N2O to the atmosphere. We take a look at this schematic here. What we see is that when N2O is, well, when it's produced in the ocean, it's eventually ventilated to the atmosphere, where it first enters the troposphere. And here it acts as a greenhouse gas with a per mole global warming potential, which is about 300 times that of carbon dioxide. After some period of time, that N2O will survive transport up into the stratosphere, where it can undergo reactions that destroy ozone. So we've got a gas that's having a two-pronged effect here in the atmosphere. On one hand, contributing to atmospheric warming. On the other hand, it's contributing to ozone destruction. And since the Montreal protocols and the reduction of CFCs, N2O has been identified as being sort of the #1 ozone destroying gas as we move through this century. So now just briefly, the production pathway. So here we've got these two major sort of encycling processes that I mentioned before, nitrification and denitrification. In nitrification, N2O can be produced as a byproduct during the oxidation of ammonium to nitrite, the first step of nitrification. Those same nitrifying bugs can also reduce, under certain conditions, can reduce nitrite to N2O through what we call nitrifier denitrification. In the more canonical or in canonical denitrification, N2O is an intermediate in the reduction of nitrate to dinitrogen gas. So it's being both produced and consumed. But under the right conditions, denitrification can produce more N2O than it consumes. So it can be a net producer of N2O. Looking right now at the sort of theoretical role of oxygen in the oceans on N2O production. So we've got N2O production on the y-axis, oxygen consumption on the x-axis. When we have fairly oxygenated conditions in the ocean, the amount of N2O produced versus the amount of oxygen consumed, which is an indicator, let's say, for how much nitrification has been occurring, we see very low amounts of N2O being produced. But as we get into low oxygen conditions, that N2O production can really increase, or the number of moles of N2O produced for every mole of oxygen consumed increases. Really, the take-home message for this is simply that as oxygen concentrations decrease, at some point, N2O production could eventually start to increase quite substantially. And this raises some cause for concern because, as I'm sure many of you are aware, oxygen concentrations in some regions of the ocean are decreasing. Oxygen minimum or oxygen deficient zones are increasing in size, and the frequency and the intensity of coastal hypoxic events is increasing. So, at some point through this century, it's likely that we're going to see N2O production really ramping up. So this is what a number of years ago when I started getting interested in N2O, and I wanted to sort of set out my research strategy. I defined it as being, well, I wanted to set my research strategy to essentially define the oxygen concentrations which turn on and turn off the different production pathways, and the oxygen concentrations or thresholds which lead to large increases in N2O production. If we can start to do this, then we can start to predict at what point through this century will N2O production really increase, and by how much it will increase, and then how much will be released to the atmosphere. So the next sort of question to address was, okay, under what kind of conditions do we want to address these topics? And by that, I just simply mean, do we want to do this in the lab? Or do we want to go out in the field and look at this in real world conditions? So as far as lab studies go, there have been a number of very important lab studies that have given us some real insights into the role of oxygen and N2O production. The first was, and I'm just going to touch on a few here. So the first was, this was the first sort of real major one, Goro et al. 1980. And they cultured ammonium oxidizing bacteria. What they found was that as oxygen concentrations decreased and got very low, N2O yields increased from about 0.03% to 10%. When I say yields or percent yield, I'm going to be interchanging N2O production and yields. Yields, I'm just referring to what percentage of every mole of ammonium that's oxidized has been yielded as N2O. So this was a massive increase. About 3 decades later, Frame and Casciotti came along, and they cultured ammonium oxidizing bacteria at concentrations, or sorry, cell densities more realistic to natural conditions. And they found that, yes, okay, N2O production does increase, but the effect wasn't as pronounced as the Goro et al. Study. However, Ellis and Santoro then showed that it's actually the ammonium oxidizing bacteria that are capable of producing much more N2O than their bacterial counterparts under low oxygen conditions. So again, this brings us back to if we're in the field with mixed assemblages, we're likely to see an increase in N2O production under low oxygen conditions. So those lab studies have given us some really important insights, but the fact is they don't reflect natural conditions. And at the time I was sort of getting into this, I would say we still, we didn't have a clear understanding of how N2O processes would respond to decreasing dissolved oxygen concentrations under natural conditions. So basically I pointed out that then we really need to start addressing these questions under natural mixed assemblage conditions in the field. So we need to go out to sea and do this. Having decided that, the next thing was, okay, where are we going to do this work? To do it, we really then need to identify coastal and open ocean regions that are characterized by well-defined oxygen minimum or oxygen deficient zones. Put another way, steep oxygen gradients, where we're going from oxygenated surface waters down to very low oxygen, even anoxic zero oxygen conditions at more intermediate depths. So we're basically using these systems as natural laboratories. Just to give you some ideas about some of the regions we've worked in. And actually, I'm going to use this as sort of segue into something I wanted to sort of highlight as well. And I think Nick mentioned this briefly last week, is that while a lot of us, all of us at BIOS, we do have a research footprint in the Sargasso Sea and the waters around Bermuda. But a lot of us also work all over the globe, in the Pacific and the Atlantic and coastal regions and so forth. And this will sort of highlight this a little here. So a couple of, some of the coastal regions that we worked in include, Saanich Inlet on southern Vancouver Island. I've worked in this region a lot. It's a great natural laboratory for sort of understanding the impacts of oxygen on biogeochemical processes. We published work from the Santa Barbara Basin using cow coffee time series data. Also worked in at Botnisek, which is a time series station run by Herman Bange at Guiomar in the Baltic Sea, just in Eckernfjord Bay here. So Kiel and Guiomar Institute is here. So very short daily trip. You can just go out around the corner and collect samples. Then what about the open ocean? So this figure shows oxygen concentrations at intermediate depths around the world's ocean. Sorry, I'm going to apologize, but the scale hasn't popped up there. But essentially, the hotter the pink, the lower the oxygen concentration. So we've got a couple major zones. We've got the Eastern Tropical North and South Pacific, and then we've got the Indian Ocean. There's something here going on here in the Benguelan upwelling system, but the oxygen concentrations still don't get very low as far as in the context of nitrogen cycling. We're not going to see any major changes. And that's true pretty much of the entire Atlantic. The oxygen concentrations are high in the Atlantic. There's an exception to this, which I'm going to touch on in a few slides' time. But let's now move on to, So yeah, just to give you an example then of one of the cruises we've operated in these regions. So in 2015, myself and my co-PI and co-chief scientist, Chris Marandino, ran the Astro OMZ cruise. So here we've got just a figure showing the track. So went north of the equator, then dropped down through Peru, or in the waters off Peru, terminating in chilling. In this case, I apologize, the scale's now flipped. So now the darker the blue, the lower the oxygen concentration. So we had funding to support about 36 scientists or PIs and technicians on this cruise. And we investigated a whole suite of biogeochemical processes in the oxygen minimum region, as well as air-sea gas exchange in the overlying surface waters. Obviously, one of the main focuses from my group was the nitrous oxide story. So those are some of the regions we work in. Now, what about the kind of measurements we make, and how do we interpret, how do we use these to sort of understand N2O cycle? So first off, very straightforward, we need N2O concentrations and oxygen concentrations. From this, we can calculate things like del N2O and AOU. Del N2O is essentially a measure of how much, or an estimate of how much N2O has been produced, excuse me, within a parcel of water since it was last in contact with the atmosphere. So we can think of it as N2O production, or how much N2O has been produced. AOU, or apparent oxygen utilization, is an estimate of how much oxygen has been consumed within that same parcel of water since it was last in contact with the atmosphere. So we can look at this now as sort of N2O production versus oxygen consumption. Let's give you an example. This is some data from the Eastern Tropical South Pacific. So we've got del N2O on the y-axis, AOU on the x-axis. What we see is that as oxygen concentrations decrease, or as oxygen consumption increases, the amount of N2O being produced versus the amount of oxygen being consumed increases. That is until we get on the right side of this dashed line. And that represents the anoxic zone, so where we've got no oxygen present at all. And denitrification is switching from being potentially a net source to now being a net sink. So it's starting to consume N2O and remove it from the water, or convert it to dinitrogen gas. We then also use N2O isotope and isotoper signature measurements. So by this, I mean looking at the del 15N and del 18O signature of N2O. And then the 15N site preference, which is the intramolecular distribution of 15N in that linear NNO molecule. We can use these measurements to start to identify how the different production or cycling pathways have contributed to the bulk N2O concentrations that we see in the water. So we can understand which processes are really contributing to the N2O that we're seeing in the ocean, and then that's getting to the atmosphere as well. So just give you an example. This is data from a paper that my postdoc, or just previous postdoc, and I recently published in Biogeosciences. And this is, we've got the del 15N of N2O on the left, the del 18O in the middle, and the 15N site preference on the right. I'm not going to go into detail about this, but I just want to sort of highlight that you can see as we get down and oxygen concentrations decrease, at some point the isotope signatures really shift. So we can start to pry apart and understand, okay, what point is do the nitrifying bugs start switching onto nitrify or denitrification? At what point does denitrification start being a net producer of N2O? And at what point does it start to consume N2O? And use 15N tracer techniques to measure the actual rates of N2O production at the time we're sampling. So we can take a sample of water in a gas type vial, let's say from a Niskin bottle on a ship. We can inject it with 15N labeled ammonium. We can trace that 15N from the substrate pool, the ammonium pool, into the N2O pool, and actually calculate the actual rate of production. We can do that under varying oxygen concentrations. You can also do this with 15N labeled nitrite and 15N labeled nitrate to look at N2O production from the reductive pathways. And just give you an example here. So this is from a paper that my postdoc Jimmy and I also just, a couple of years ago, published in Journal of Geophysical Research. This is from Saanich Inlet. And we've got nitrification yields. So remember, this is the percent of ammonium that's turned into N2O. This oxidized ends up turning into N2O. But nitrification yields here, we've got the oxygen concentration during the incubation period on the x-axis. You can see that the yields, when we've got oxygen concentrations going down to about 20 micromoles of O2 per liter, the yields are pretty low. We're talking around about, let's say, 0.05%. But as soon as we drop below about 20%, 20 micromoles of O2 per liter, the N2O yields skyrocket. We're getting up to sort of now about 4% yield. So this is again sort of just demonstrating that, you know, the kind of oxygen thresholds where we see these large increases in N2O production. And finally, I don't make these measurements myself, but through collaborations, we also look at the abundance and the expression of genes associated with N2O cycling. So when are these turning on and turning off? And then finally, I use microsensor measurements from microelectrodes to look at investigate N2O cycling in marine sediments. And I'm working largely with, so I'm an adjunct member of the Faculty of Graduate Studies at the University of Victoria. One of the students I'm co-advising, Brett Jameson, has been using my microsensor package to look at N2O cycling in marine sediments. And this is just an example of one of the projects we ran. This is in the continental margin off of Vancouver Island. So we went out on the ship with a submersible. We went down and we collected sediment cores at various depths. We brought those cores back up to the surface. It was an autonomous vehicle. We weren't actually physically in it. And we incubated those sediments in chambers with flowing seawater for periods of time. And every so often we could take those cores out and we take a microsensor, which is a very minimally invasive approach. I don't know if you can see it very well here. It's a very fine sort of needle, goes through the sediment core, very high resolution, vertical resolution, and we can measure N2O concentrations and oxygen concentrations. We can then apply curve fit modeling techniques to those results, and we can estimate how much N2O is being produced, how much is being consumed, how much is getting out from the sediments to the water, or vice versa. And in this case, we found that the sediments were a very important source of N2O to the water column. We're just wrapping up another paper where we did this in pristine mangrove sediments around Bermuda. And in that case, we found that those sediments are really important sinks of N2O. So they're drawing N2O out of the water column, which provides another argument for let's leave these mangrove regions alone. Okay, so this gives you an example, idea sort of the places we've worked, type of measurements we use. Before I sort of finish up though, I wanted to at least take you through one story from start to finish. And I picked this one because one, I think it's a pretty cool, it was a kind of pretty cool new finding. But two, I think it's a pretty straightforward story to take you through in a few steps, especially for anyone that's not into the nitrogen cycling sort of process. So this is a paper that we published titled Low Oxygen Eddies in the Eastern Tropical North Atlantic and Their Implications for N2O cycling. So just give you a bit of background. So back from about 2006 to 2013, colleagues of mine at Guiomar had a mooring out at the Cape Verde Ocean Observatory, just northeast of the Cape Verde Islands here. On that mooring at about 50 meters depth, they had an oxygen sensor. And when they compiled all that data, what they found was that, and remember I said the Atlantic Ocean is always really well oxygenated. This is the exception. So when they compiled all this data, they found, highlighted in gray here, that every so often, oxygen concentrations just drop down to, close to or even to zero oxygen concentrations. What they found was that every time one of these events occurred, it corresponded with an eddy passing through the region. So eddies are these circulating water masses that propagate through the ocean, and they can have very distinct chemical and biological signatures. And this, let's just see if this is. But here you can just see these eddies. They're essentially forming off the Mauritanian upwelling system. They hook off here and start spinning, and they propagate past the Cape Verde Islands. So the conclusion or the thought was that some of these eddies must be characterized by really low oxygen conditions. So when I got to Guiomar in 2013, a few of us wrote a proposal to the Future Ocean Cluster of Excellence, and we got awarded to go out and conduct a short eddy hunting survey and collect some, make some measurements for different things. So in late 2013, we started using satellite imagery to identify a potential eddy candidate, low oxygen eddy candidate. Then in early 2014, we went down to Cape Verde. We deployed a couple of gliders. We were able to drive these gliders into this eddy candidate. One of the sensors that was on that glider was an oxygen sensor. So we were able to, instead of having to go out on a ship and back and forth trying to find the eddy, we were able to just use these gliders to go out and do the job for us. And you can see here, so we've got temperature, salinity, oxygen, and nitrate. What I really just want you to focus on here is the oxygen, the third figure from the left. And when we look in the center of this figure, we can see that we're getting this pool of really low oxygen water in the core of the eddy. So this confirmed to us that, yep, we've got a low oxygen eddy. So now in March 2014, Bjorn Fiddler and myself went down to Cape Verde to go out and do some shipboard sampling this eddy. This was anything but trivial. We were using a very old, very broken down Icelandic fishing vessel that had been donated to the Institute there. When I first got there, I'm looking at the ship, the fenders are falling off, the ship's rubbing up and down as the concrete dock, bits of it are falling off. It took us a number of attempts. We were down there for like 4 weeks just to try and get out for a day or two of sampling. We finally managed to do that. And we were looking at things like vertical migration of zooplankton. How are these dead zones or these low oxygen zones impacting that? We were looking at some of the physics, the carbon chemistry, and then of course, nitrous oxide. So just give you show you some of the results here. So got dissolved oxygen through the water column on the left, N2O through the water column on the right. Black lines and black data points represent a reference station outside of the eddy. So our kind of typical conditions that we see in this region. Red represents a couple of the stations inside of the eddy. So we can see, again, here, the oxygen concentrations in the core of the eddy are really dropping off. You know, one station we were down to just under 10 micromoles of O2 per liter. At another, we were down to just under 5 micromoles of O2 per liter. Looking at N2O, outside of the eddy, the concentrations are pretty much bang on what we see in this region. And really anywhere in the Atlantic, they don't get much higher than about 40 nanomoles of N2O per liter. But inside of that eddy, we were getting, they just ramped up in the oxygen minimum zone. We were getting up to close to 120 nanomoles of N2O per liter. And this is by far the highest concentrations that have ever been measured in the Atlantic. And they're on par with sort of the high N2O concentrations that we see in the Pacific and the Indian Ocean. So again, we calculated del N2O versus AOU. And we found we had two very distinct linear relationships, which I've just called relationship 1 and relationship 2. So this first one is at oxygen concentrations above 20 micromoles of O2 per liter, about 22 micromoles and above. We can see we're getting, not a lot of N2O being produced versus oxygen consumed. But as soon as we drop below 20 micromoles of O2 per liter, that N2O production really starts to ramp up. And if we look at the slopes of the relationships, we were able to say that above about 20 micromoles of O2 per liter, 8,500 moles of oxygen would need to be consumed to produce 1 mole of N2O. Below 20 micromoles of O2 per liter, we were able to say that now we only need 170 moles of oxygen to be consumed to produce 1 mole of N2O. Put in sort of maybe simpler terms, that's essentially a 50-fold increase in the amount of N2O being produced versus the amount of oxygen being consumed. As soon as we hit this threshold, it's a very sort of abrupt tipping point that we're seeing. And so in addition to providing some information, some new information for the Atlantic Ocean to say, hey, it's not quite as boring in terms of N2O as we thought it was, and we need to start taking these eddies into consideration. But it's also, again, we're using it as a natural laboratory to provide that sort of first insight into the potential N2O conditions that we might see in the tropical Atlantic Ocean decades from now as we move through this century if oxygen concentrations continue decreasing. And very briefly touch on some isotope results here. So we've got del 15N, del 18O, and the 15N site preference of N2O. Red is inside the eddy, black is outside of the eddy. And what I really just want to focus on here is these circled points where we saw really massive changes in the isotope signatures. And this indicated to us that denitrification had started to become an important source of N2O. And I'm just highlighting this because I think this is kind of pretty cool as well. This is the first time that we've ever seen any evidence for denitrification, water column denitrification in the Atlantic Ocean. So again, these eddies are pretty cool features to get out there and study. It's not easy, but it's certainly worthwhile. Okay, so just to wrap up with a few thoughts on concluding points. So not just in low oxygen eddy, but from the work that we've done, in other regions, in coastal regions, Atlantic regions, in the Pacific, and that colleagues of mine have also worked on. I think we now have a much better understanding of marine N2O budgets and the current rates of production in different marine regions. And that these low oxygen regions that we've been studying really are ideal natural laboratories for understanding how N2O production could respond to future marine oxygen conditions. So I'm going to generalize a little bit here. It does vary slightly from region to region, but I think we can now begin to better constrain the oxygen thresholds, which induce substantial increases in N2O production and cause major shifts in the production and consumption pathways. So I think in a general terms, we can say that below about 20 micromoles of O2 per liter, N2O production really starts to increase. And you've seen how much, just by how much it can increase. Below about, but it's still nitrifying bugs are the ones that are really producing it, whether it's through ammonium oxidation or nitrifier denitrification. Below about 10 micromoles of O2 per liter, denitrification becomes an important net source of N2O. And then somewhere between, that's a bit below 5 micromoles and 0 micromoles of O2 per liter, denitrification becomes a sink for N2O. So I think these results that myself and other colleagues have been coming up with really are useful. We now need to start using them to inform the biogeochemical models. People that are sitting there trying to predict what's going to happen with oxygen in the future. We can now start plugging in some of these findings into those models to start predicting how much N2O will be produced or when's the tipping point going to occur. It's not quite as simple as that. There's a lot of complexity there. We've got to think about feedback mechanisms. You can imagine if we've got more N2O going to the atmosphere and the atmosphere is warming more, then we're going to have more stratification. We're going to have less oxygen penetrating down to depth. Consumption is going to speed up. And so we can think about these in multiple different ways, but there's a lot of complexity here. So it's not just as straightforward as plugging this into a model, but you see where I'm going. Okay, so just now spend the rest of my time just thinking about some future research plans. So just because I said we've found some really cool results so far and we've got a much better understanding of N2O production, doesn't mean we should stop looking at this. We need to continue this in other regions. We need to start looking at other environmental variables, quality of DOC, for example, dissolved organic carbon. I will say, could you imagine if we stopped measuring primary production in the 70s? We just wouldn't have the same understanding as we do today of primary production in the ocean, and we wouldn't have been able to sort of track those changes as well. So we do need to continue with this work, and we will continue with it. One of the other things I'm really interested in here is that a lot of us have been working on this topic of N2O production in the ocean. But we do have N2O consumption in anoxic zones occur. So you can imagine if these hypoxic or low oxygen zones in the ocean expand, we get more N2O being produced. But what if the anoxic zones, where we've got denitrification consuming N2O, also expand in size? It may act to buffer, at least to some degree, against those increases in N2O production. Trouble is, we really don't have much of an understanding about this. So I wrote a proposal to NSF that was funded recently with my co-PI Frank Stewart, and we're going out trying to investigate this. We just recently finished our first sampling campaign in Sandwich Inlet, getting back to Sandwich Inlet. It's just an awesome place that is easily accessible. It's got predictably low oxygen concentrations. So we've been out, we've started this, and just want to show you some very preliminary results. I just got these a few days ago. This is from just one of our sampling days in Sandwich Inlet at just 2 different stations within the inlet. And I'm realizing that the depth hasn't shown up here, but basically we're going from the surface down to about 200 meters depth. So when we've got oxygenated water, even at the hypoxic anoxic boundary, we've got no N2O consumption occurring. But as soon as we get into the anoxic zone, we see N2O consumption really ramping up. And basically, just doing a quick calculation in the last couple of days, I think the kind of rates of N2O consumption that we're seeing, they could certainly consume any N2O that's diffusing from the overlying waters down to the anoxic zone about as fast as it's coming up. So this is certainly giving us some idea that denitrification in anoxic zones can act as a buffer. We don't know to what degree yet. That's going to be worked on over the next sort of 2.5 years still. Another topic which I'm interested in is this idea that sulfide can inhibit N2O consumption. And I'm working on a proposal that will be submitted to NSF in about 2 weeks' time to look at this. So the idea is that, okay, we've got this where it just adds layer to layer of complexity, right? It becomes a bit more complicated. So we've got these low oxygen zones where we've got N2O production occurring. We've got anoxic zones where denitrification can consume some of that N2O that's been produced. But in anoxic regions, at times we see sulfidic events occurring. So if that's preventing denitrification from consuming N2O, we need to understand this as well. And just to give you an idea, this isn't the only evidence, but just a quick figure that I put together over the weekend. This is N2O consumption on the y-axis, hydrogen sulfide concentrations on the x-axis. But we've got these very low sulfide concentrations. This is basically the hypoxic anoxic boundary, where we don't have N2O consumption occurring anyway. We get into the anoxic zone, N2O consumption really increases. But then when we get above about 5 micromoles of H2S per liter, we see that N2O consumption slowing down. It's not completely inhibited, but it does seem to be slowing down. And just so you know, it's not because there was less N2O. The N2O concentrations were, we actually experimentally manipulated and kept them all sort of the same. So, you know, that's another topic that I think is worthwhile. And again, I'm working with Frank Stewart on this. Frank is my collaborator. I work with a lot for the molecular genetics side of things. So we're hoping if that was to be funded, that would start up early next year. Yvonne Saval, myself, and our co-PI, Natalie Goodkin, were just awarded another NSF grant to start looking at the impacts of eddies on Bermuda coastal chemistry and coral performance. So again, we've got these circulating water masses that have fairly distinct or you know, different chemical signatures. What are those chemical signatures? Is those eddies sort of hit Bermuda, basically. And then what's the, from Yvonne's side, what's the impact on the corals? I think she'll talk about that a bit more tomorrow. And then a couple other ideas. I've got, you know, there's a number of other ideas that aren't just nitrous oxides. I figured because the talk was nitrous oxide, I'll stick with this. But looking at how N2O cycling in the Sargasso Sea surface waters could be impacted by things like microplastics, where we see a lot of microplastics in the Sargasso Sea off Bermuda. And then we also have these large sargassum seaweed patches, hence the name, the Sargasso Sea. So, you know, these kind of things like microplastics and sargassum, are they able to sort of create these conditions where you can sort of get these buildup of biofilms of the sort of bacteria in the archaea that are producing N2O? And do we see suboxic microzones forming where we might see more N2O being produced? I've gone out and I've taken N2O concentration measurements in the Sargasso Sea. They're very low. But the point is, if it's right at the surface where these things are promoting N2O production, we're not going to see it through a concentration measurement because this is going to be ventilated straight to the atmosphere. So I've been talking to Rachel Parsons, who's a senior research technician at BIOS, who's had a lot of, you know, done a lot of work with microplastics and seaweed, and is really interested in this, looking at the molecular side of things. I've also been talking to Suzanne Neuer about microplastics. Suzanne has a lot of experience working with microplastics, so we're planning to meet in a couple of days to discuss potential proposal that we'd like to put together. And, I think the thing that we need to do is really sit down and think about the experimental approach here. How are we going to identify whether there's N2O being really produced without, one, disturbing these systems too much? I think it's going to be a combination of lab and field studies in this, where we're going to need to go out and collect, incubate, and get back to using the 15N tracer techniques to look at this. So, with that, I can wrap up, and well, it's up there, better acknowledge my funding sources, so everything from sort of NSERC in Canada to German Research Foundation and the Ministry of Education and Research, which funded our Astro OMZ cruise, the Future Ocean Cluster of Excellence in Kiel, SFB 754, and then obviously the National Science Foundation as well. And I'm happy to take any questions.
If anyone who's online wants to type some questions in the Q&A, and then you just go ahead and field questions here in the room, and I'll look out for the Q&A and read those the online questions.
Great.
So thank you for a great talk.
Oh, thanks.
We don't get deep dives into biogeochemistry around here very often. I really appreciate that. I have a simplistic, and it's actually probably a naive question, but a lot of the N2O production data you showed us was plotted against AOU. And the tipping point you made clear over and over again is when oxygen concentrations are extremely low or not, but that's when AOU takes off. How do you have apparent oxygen utilization when there's no oxygen?
Yeah, so it's, let me just see if I can pull up that. So we're not really getting any more AOU, but certainly if we get a bit of sulfide, it can start to get negative oxygen concentrations essentially, which will show up in a higher AOU.
Negative oxygen concentration?
If sulfide is present, you start to kind of get into a position where you need more oxygen just to get back to zero, because the sulfide will just sort of poison that oxygen. It's not something I've just, somebody was just explaining that to me a while back, but yeah, I mean, some of this is also just due to probably some error and a bit of diffusion. So some of this might not strictly all be the anoxic zone. Some of this could be nitrifying N2O consumption signatures that have actually diffused into the boundary, the hypoxic anoxic boundary. So maybe the anoxic zone is really sort of right here where we don't see any more oxygen consumption occurring, but we're really seeing the increase, the drop off of N2O. Yeah. All right, I'm not sure who was first there.
How much of marine N2O production comes from anthropogenic nitrogen deposition from the atmosphere and discharge in rivers? Is that causing an increase?
Not really any. We do have anthropogenic N2O release from soils, from industry. I mean, some of it you could say is anthropogenically derived if you're pumping ammonium into a system, and that's creating an anoxic condition, and then, or a suboxic condition, and then you're getting N2O produced. You could say that's anthropogenically derived, I guess, from the standpoint of the substrate has started off as an anthropogenic source or a pollutant, if we get into coastal regions. But yeah.
But the mechanism is mostly the impacts on oxygen concentrations rather than an increase in N sources.
Yeah. Well, that said, I mean, no, because if you're in a coastal system and you're really loading it up with ammonium, you're going to get more phytoplankton growing. You're going to get more organic matter settling out. You're going to have more bacterial consumption of that organic matter, more oxygen consumption. So you're going to add, you are artificially driving down the oxygen concentration.
Right. We have another question. It's been said online that they can't hear the questions very well. So after you ask the question, perhaps you can repeat for the online audience to hear.
For your work in like Peru, have you guys looked at El Nino and La Nina events impact on
Yeah.
N2O production? And
Actually, we were, so in this, I didn't want to get it, I wasn't going to get into it just because it wasn't sort of the time, but this cruise that we ran in 2015 happened to be just as an El Nino event was really starting to ramp up. And we've published a couple papers on this. So one looking at the sort of the impacts of the El Nino had on nutrients, but also on the impact that it had on nitrous oxide. And let me just think about which way around this went. So what we were finding was that there was less, the anoxic zone wasn't as well defined as it typically is. So we were getting more N2O. Wasn't necessarily that the N2O production rates were higher, but the consumption was less. So when we had this El Nino event and compared it to previous cruises that we've had in this region, we were able to show that we actually had more N2O because we were not getting as much consumption. That zone of consumption wasn't as large as it was. So,
Back to the question about oxygen utilization. Are you strictly working with utilization, or are you also looking at the rates?
Well, we do sometimes look at rates, but more, not in great detail. It's more to kind of confirm that we're not getting, let's say, large changes in oxygen over the course of a several hour incubation. But no, we're just taking oxygen concentrations and calculating, an AOU from that.
We have something online. Hillary Hartnett would like to say thank you for a lovely presentation. And she wonders, do you see N2O consumption all the way to the sediments in Saanich, or only just below the oxic/anoxic interface?
No, we see them all the way down. I'm still, and it's certainly going to be occurring in the sediments in Saanich Inlet. I've been taking samples from close to the sediments. I'm not sure what to do with them yet. I want that data, but I'm not sure it's going to be very easy to incorporate into this story of using Saanich as sort of something that we can extrapolate our findings to other regions. Because if you've ever been down in Saanich Inlet, the sediments start 10 meters before the actual gets hard. It's like just a soup. And, so I'm trying to avoid getting any contamination of samples for one. And two, I don't want to, confound results from N2O signatures that are coming out of the sediments with what's actually happening in the water column. So we are taking that, but I'm not going to necessarily use it as much in just identifying what's happening in the water column.
We have another one that says, great talk, Damian. What are your thoughts on future trends of N2O production in a world of declining oxygen concentrations in the ocean?
Yeah, so I certainly think, we're going to hit a point at some point through this century. This is something I need, I really need to sort of maybe sit down with. I don't want to start giving you a year or a decade. I work more with the biogeochemical modelers for that, but I really think we're going to start to see an increase, a very large increase at some point, and that's going to get out to the atmosphere. But we then need to think about feedbacks that we might see. As I mentioned, on one hand, could be a positive feedback. So we might see more N2O in the atmosphere, warmer conditions, more stratification in the ocean. So even less sort of oxygen, penetration and things like this, which could lead to even more N2O production. On the other hand, we could end up with a negative feedback system where we because of that stratification, we're getting less nutrients coming up to the surface. We're getting less primary production occurring, less organic matter going down. So now we've got less oxygen being consumed. I think this is a very complex sort of thing to look at, and it really needs some hardcore modeling techniques to be done. Peter.
Yeah. Go extrapolating forward. So if you take your best guess of what an extreme situation of the increase of N2O production in the ocean could be by whatever, 2050, 2100, you could pick the time. Then which fraction of the greenhouse gases would actually be N2O rather than CO2, CFCs, etc. So in other words,
Yeah.
how big will the effect actually be? Is it a few percent? Is it shifting? You know, we typically say 2/3 or so is CO2, and then we have methane and CFCs
Yes.
and N2O typically. These
Yeah.
are the main ones. So what could the envelope be in which N2O might reside in the future?
I'm not sure I'm going to commit to an answer on
And
that.
I'm not looking, I'm just looking for some, I do understand how early it is in
Yeah.
the research, but if you would say, what is your best guess, or you could even call it speculation, but just to get a sense, how
Yeah,
how
maybe
much are we actually expecting here?
maybe up towards maybe something like 5 to 10%. That's just thinking, running back of the envelope in
Yeah.
on the fly there, because I'd also have to think about how we're expecting methane to change as well, because if methane's also increasing, then it's going to keep dwarfing the impact of N2O. But the N2O is not certainly the most important one out there, but it is having contributions. And it's also, but it's going to be the #1 ozone destroyer for sure.
And that would have been the next question, but
Yeah.
yeah, thanks.
What if North Atlantic deep water formation slows down? Like, how do changes in ocean circulation affect global marine N2O emissions?
Yeah. If information slows down, we're going to have less oxygen penetrating down to depth and being transported. I mean, it's one of the reasons why the Atlantic just doesn't have very low oxygen at the moment, because we have that deep water formation starting off in that region. So certainly that could, reduce this. It's going to reduce the supply of oxygen from surface waters down to intermediate depths, which is where a lot of the N2O is actually produced, in the open ocean. You can also think about it as, there's other interesting things is sea ice formation. We're getting much less sea ice formation and much more. Let me just see if I can quickly. No, it's on my computer. We're getting much less sea ice being formed, and this is really slowing down the supply of oxygen. When sea ice is formed, you get brine rejections. This very salty, heavy water just is taken out of the ice and it sinks down, taking oxygen down with it. So that's, we're already seeing that happening in the Atlantic. We see it a lot in the Northeast subarctic Pacific. So there's a time series station there, Ocean Station Papa, where I've worked a lot in the past. And, from the 50s, they've been looking at oxygen concentrations, and they've been decreasing. It decreased by probably about 25% now at intermediate depths. And that's going to speed up, because what happens is over in the Sea of Okhotsk, I can never really pronounce that, over in Russia, you get a lot of sea ice being formed. Well, typically, we're getting much, much less sea ice being formed. So less oxygen is getting down to those intermediate depth waters, into the intermediate depths, which then transit across the Pacific over towards the Northeast subarctic Pacific. So we're getting much less oxygen being supplied. So we're just not starting off with as much oxygen as we typically would have.
So just to clarify, when you're talking about sea ice formation in the Atlantic sector, you're probably looking at the area south of the silts of the Greenland Iceland,
Yeah.
Scotland silts, so Ermine Sea and Labrador Sea.
Yeah.
That's like a lot of positive feedbacks.
Yes, but I think there are, certainly at some point you're going to get to a negative feedback. You imagine if we're getting, so think about it from another way. If we're getting less oxygen, we're getting more denitrification occurring eventually. And so we're going to have maybe consuming N2O, but we're certainly going to have a lot of dinitrogen gas being formed. That leaves the ocean and just strips the ocean of bioavailable nitrogen. So now we just don't have any phytoplankton production occurring. But now we've got less CO2 being taken up from the atmosphere, or by phytoplankton. So there's a lot going on there. That's why I say we shouldn't be drawing a line under this and saying, where do we go? What's the next thing we want to work on? It's
Could be busy for half a century.
yeah.
I don't have any other questions online. Any others in the room?
So one final.
Yeah.
So which fraction of the work that you're doing do you think you can do through gliders? And which fraction still needs conventional seagoing?
Yeah, let's say if most of my work is nitrogen cycling, in terms of oxidative versus reductive nitrous oxide, we're just not there with gliders. There's, we could use, there are gas tension devices that we can put on the instruments to look at things like denitrification or signals and things like this, which I believe you've worked with quite a bit. There's certainly other automated things, underwater submersible mass specs. I think is a pretty cool idea. I even think that you could take the Picaros and probably build an underwater pressure housing. And I've been thinking about that quite a bit. I still collaborate a lot with folks in Victoria, University of Victoria and Ocean Networks Canada. And I'm not sure if anyone's familiar with Ocean Networks Canada, but it's basically a cabled observatory system that goes from Saanich Inlet and the coastal regions on Vancouver Island all the way off the continental shelf and down to the abyssal plains. It's a great place to test out instruments because that you might want to adapt. You could go into Saanich Inlet, you could take a submersible mass spec or a Picaro, but put into a pressure housing, waterproof pressure housing, and you don't need to worry about batteries. You just plug it into their power and just have it sitting down at, let's say, 120 meters depth and seeing what's going on. So there are other sort of autonomous kind of things I think we could be using more and should probably start thinking about the next decade. But I think gliders were probably still fair ways off of that right now.
Well, we are coming up on time, so it's kind of perfect. We have no more questions online as well. So thank you very much,
Great.
Damian. We really-
Thanks.